This video tutorial will demonstrate the proper assembly, setup, and interpretation of the respirometers you will use in the Cellular Respiration Lab. It is important that your respirometers be assembled correctly for them to provide you with accurate data. Except for the peas and beads, each respirometer is assembled the same way. Respirometer assembly starts with creating a tight seal between the pipette and the rubber stopper. Take your strip of parafilm, place it approximately 1.5 inches from the blunt end of the pipette, and tightly wrap it around the pipette. Parafilm is stretchy, so gently tugging on the parafilm as you wrap it will help the film adhere to the glass. Holding the pipette by the wrapped section, carefully insert the blunt end of the pipette through the hole in the stopper. Do not shove the pipette into the stopper abruptly or hold the pipette too far from the stopper, as this may cause the pipette to break and cause cuts or puncture wounds. To make sure the seal on the stopper is airtight, plug the top of the vial with the stopper and completely submerge the respirometer in the water bath for 20 to 30 seconds and check to see if water is entering the vial. If the vial is leaking, this may be due to poor contact between the stopper and the pipette, or between the stopper and vial. Simply tighten the stopper and/or the pipette to correct this. Be sure to check all 3 respirometers for leaks. It is better to detect and repair any leaking respirometers at this point before the experiment has begun. If your respirometers leak during the experiment, you will need to restart the experiment from the beginning. Follow the steps in Activity 1 of the lab manual to assemble the contents of the respirometer vials. To complete your respirometers, food coloring must be added to the pipettes. This will help you observe volume changes in the pipette during the experiment. Carefully lay the 3 assembled respirometers on a flat surface with the pipette tips positioned over a paper towel. Add some food coloring to the tip of the pipette by hanging a drop of food coloring from the bottle so that the drop just touches the tip of the pipette, and allow capillary action to pull the drop into the pipette. This will color any water that enters the pipette, so the demarcation between the air and the water in the pipette is easy to see. Now that your respirometers are assembled, they will need to equilibrate in the water bath. Begin this process by placing the 3 respirometers into the water bath in an upright position, the pipette tips resting on the lip of the bath container. If any of the respirometer vials begin to fill with water, you must restart the experiment. After 5 minutes, submerge the respirometers in the water bath. Position them so that the scale on the pipettes can be easily read. You will notice that as a small amount of water enters the pipette and is colored by the food dye, some dye may escape into the water bath. Your pipettes will be marked with 2 different scales. Use the scale that reads 0.9 at the tip and 0 toward the stopper. After you make these adjustments, do not put anything into or take anything out of the water bath until the experiment is complete. The purpose of the respirometers in this experiment is to show changes in volume caused by any cellular respiration occurring in the vials. When the respirometer is submerged, part of the pipette fills with water, while the rest remains filled with air. The volume of air in the respirometers is measured by noting the point to which the water level reaches within the pipette. For example, in this pipette, the water level ends here, which means that there is 0.5 milliliters of air in the pipette. Calculating the difference between the volume measurements you take over time will give you the change in volume in the respirometer.